In this video, we're going to talk about the biology of a maggot mass, arguably one of the most important things to understand when doing case reports, because majority of the time we're working with maggots coming from maggot masses. So it's very, very important. Now, when I say maggot, I'm referring to fly larvae. It's very rare that you use it as anything other than fly larvae, but it is just colloquial terminology. So in things like reports, we'll say fly larvae, but we will just call it a maggot for the purposes of this class. So when we're talking about necrophagous flies, so things like blowflies, flesh flies, house flies, all of them are what we call gregarious, meaning that they rely on more than one individual to do at least one part of their life cycle. So adults will rely on gregarious aggregations to find carcasses to lay eggs. In maggots, they rely on cooperative feeding to consume actual tissues. So cooperative feeding means that they are working in tandem to actually break that tissue down and ultimately consume it. So everyone kind of gets a little piece of the pie, but they're all working together. And so when you have this cooperative feeding, you'll end up being able to deplete a resource very quickly and share that resource across the maggot mass more or less equally, though some individuals are going to die because they're not going to get that resource. There are going to be specific digestive enzymes that are going to be secreted to help that cooperative feeding, right? The goal is to break down those tissues. When you have multiple individuals, not only is that tissue being broken down, but it's also being taken up very, very rapidly. So again, some individuals are going to actually lose out because while they're also contributing those enzymes, they might not make it to the carcass itself in time to actually feed on the tissues. You'll also end up generating a lot of heat. So we're going to talk about that in a little bit more detail. But because you have all this metabolism, they're actually heating up. And then you'll end up having really rapid growth rates. Oftentimes you'll have fewer molts, depending on the insect species, but we still have what's called ecdysis, which is when they're shedding that exoskeleton. So that isn't reduced, but there are minor molts that could slow down because you have all this feeding and your body is just going to develop in a different way. There are different types of maggot masses depending on how many species there are on remains, but also how or where they're aggregating on the remains itself. So something called a homogeneous mass just means that the maggot mass itself are comprised of all maggots from the same exact species. So they can have different mothers, but they don't have more than one species within it. Where heterogeneous masses are the opposite, so it can have two or more species that is located. So you can see on this picture here on the left that there are two different species that are commingling, and you can tell because they're very visually different. So this is actually two different species that where this one here, is predatory, and this one is not. And so they're actually trying to get away from that other species. So it's usually not this distinct, but it can be, just depending on the species that are involved. And usually when we're talking about maggot masses on remains, it will be a heterogeneous mass. Unless you're really early on in decomposition when the individual was found. Most of the cases I work, they're third instar, so it's pretty late in the game. But the other consideration could be barrier to entry, so if it was indoor or not, or sometimes species of carcasses, but a lot of times the carcass size specifically really plays into how many larvae are actually there. When we have the actual maggot masses, majority of the species are going to be of the blowfly family. So that's going to be comprising a lot of different species, but usually it's multiple blowflies that are on, maybe with one or two other groups associated with them. So the way that larvae actually form masses is more or less unknown. It is something that we can observe to happen, but we don't have the actual foundation to say exactly what is going on. And so there's a couple different ideas. So one is that females lay in clusters, and therefore the larvae will just emerge and hang out by their cluster. However, when you look at a carcass and you have first instar larvae, they actually don't form a maggot mass in that first instar. They actually start moving all around the resource. And so it's really easy to tell the difference between a first and second instar, because it's really based off of, one, they're super teeny tiny, but also they're going to be either extended across the resource or in an actual mass itself. So that it's a good theory, but it doesn't really hold. The other thing is we have thigmotaxis. So this is your sensation and response to touching things. So the idea is that larvae can kind of move around a resource. So again, they're doing this as a first instar. They're moving all the way around a resource. Once they start molting and they go to their next instar, they end up bumping into one another. Their cuticular hydrocarbons then touch, and then they start forming their mass together. So while this is helpful for one or two larvae to actually locate each other on the carcass, it doesn't help us understand why maggot masses form where they do on the body, because it's pretty consistent across the type of carcass that you'll have. And so while these two things are helpful, it could be a little bit of both, or, and more likely, it's something that we don't fundamentally understand yet. So a combination of the following is what is currently hypothesized as why we have mass formation. So again, it's that clustered oviposition or larvoposition. Then this would be facilitated by pheromones, so aggregation by adult females to lay and oviposit in that area. And then you'll have random formation, and usually this can result from direct competition between individuals. And what they're actually doing is looking for the exact same resource in one location. And so it could be that the part of the carcass that all the individual larvae want is going to be where they all go, and as a result it's a mass. It's not intentional, and it's not because the individual's helped by it. Rather, it is just a secondary consequence to that. And then there is this idea of larval foraging. So foraging is when you go out and you look for different food resources. And the idea is that there is a chemical attraction to the carcass, right? We know that from a female. But the idea is also that when you have species or individuals of the same species, there's going to be a heightened attraction, and this ends up extending into the larvae, where the larvae are going to be more attracted to locations where other conspecifics or individuals of the same species are. Again, there's issues with that when we talk about heterogeneous masses, but again, these are just kind of likely explanations, but again, we don't fully know yet. we do know is that these feeding aggregations do provide a benefit to individuals. When you take a maggot and you put it on a new resource and you let it feed, it will die 99% of the time. And that is because the actual physiology and anatomy of a maggot is not made to be able to feed like that on its own. So maggots have these modified mouth hooks that allow them to scrape, and they secrete digestive enzymes, but that in itself is not enough to break down tough tissue like organ tissue. And so it's necessary that you have more than one individual to help kind of overturn those tissues. So we know for a fact that even though we don't know why maggot masses are forming, we know that they're helpful. This is what fly mouth hooks look like. So let me see here. These are the eyes of the maggot. So you can see them right there. And then these are the mouth hooks themselves. Now, a lot of species you'll end up being able to identify with mouth hooks because they have different articulations on them. But all these mouth hooks, regardless of their morphology, are used for extraoral mastication, which is just chewing, right? Or most of the time it really is tearing because they don't have the ability to actually crunch on their food. So they're virtually scraping the tops of the food and then adding those enzymes to help break it down so they can ingest it. As mentioned multiple times in this point, larvae produce a whole array of enzymes. What exact enzymes they are producing is not well studied, but we do know where the enzymes are being produced. So they are produced in the upper digestive tract, so both in the salivary glands as well as the foregut and the midgut. And you can consider the foregut and midgut as like the small intestine of a maggot. We also know that while you have larger maggot masses, you're going to have more enzymes, and then you're going to have faster breakdown of the tissues and faster consumption. The issue with this is while we understand it, you can observe it, no one's really empirically tested this. So we know it from observation, but we don't have a way to actually quantify that. Now that we've covered the actual attempt to break down that tissue, once that tissue's kind of softened up, then you have to actually ingest it. So food assimilation is just the actual integrating of those nutrients into larval tissues. So actually using it for growth. So the ability for food to be assimilated at the individual level depends on metabolic rate, how much enzymes that you are producing, plus all the other individuals around you, and the optimal aggregation. So the larval mass that you're working with has to be perfect for the food assimilation. You can't just go more and more maggots because there's more and more enzymes. There is kind of like a bell curve to these things. So you need to find your optimal. So when we're talking about temperature, this is really well understood. You probably have heard these terms when you took your intro bio or if you took like an organismal biology course. Insects are poikilotheric ectotherms. And so what that means is that they don't generate their own heat, so they're ectotherms. And poikilothermic means that they can live across a wide range of temperatures. And so when you have something called heterothermy, you're actually switching between regulating your own temperature and relying on the surrounding environment. Where homeothermy is just that maintaining your stable internal temperature. So humans are homeothermic, endothermic, homeotherms, because we regulate your own temperature. We exist within a very specific temperature range, otherwise we die. And our temperature regulation is specifically internal only. So there is no switching between that surrounding environment as a reliance on temperature control. When we're talking about maggot masses, they are polychiothermic ectotherms that are heterothermy. So it's interesting because while the insects are ectotherms, when they produce metabolic heat, they are in a way regulating their own temperature, just not in the means of an internal mechanism like we have. The maggot mass is really interesting because the metabolic heat generated can be really profound. So there are most citations that say that a maggot mass can produce a temperature about 14 degrees C above the ambient temperature experienced, but there are other cases where they can be seen as high as 30 degrees Celsius. So this is really helpful for two reasons. One is that if it's really hot out already and the maggot mass is producing this heat, you might have a massive maggot die-off, which if you come onto a scene and you have all these dead maggots, that is one explanation. When you have cases in the middle of winter and someone gets colonized inside a structure, so maybe in a home, and then the maggot mass develops and then they were placed out in the snow, you can still have maggot activity on the remains. Also, if you have those remains and you take them into the medical examiner's office and you put them in the freezer, the remains, the maggot mass is still going to be active, right? So understanding that they can sustain their own temperature means that we need to take steps, right, to mitigate that if we're going to start taking evidence into places, even if we think cold storage is going to help. It doesn't. And what we see is that with increasing maggot mass, you'll end up getting increasing temperature produced, which isn't super profound, right? Of course, if you're going to have more individuals, they individually are going to generate more metabolic heat, and so it is summative, right? It just adds on top of each other over and over and over again until the maggot mass dies. Now, of course, there are benefits and challenges to metabolic heat, but primarily we want this metabolic heat as if we were larvae, because it allows us to experience increased enzymatic activity, which ultimately allows us to assimilate food into our tissues much easier and much faster. So there's higher efficiency in that. The issue with it is something called proteotaxic stress. So proteotaxic stress is basically a physiological stress mechanism that happens when you hit either the critical thermal maximum or minimum. So the critical thermal maximum is the maximum in temperature in which a organism will survive. Anything beyond that, they die. And then the critical thermal minimum is the opposite. So it's the lowest temperature that can be experienced to live. Anything beyond that, they end up dying. And then the thermal tolerance is basically in between that. And it's not just a standard bell curve as to what is the preferred temperature. So here you can see that this is a performance curve. Usually these are designed as performance curves. So you have this optimal window for whatever performance you are looking at. So if we wanted to look at enzyme activity, then we'd have the critical thermal minimum where enzymatic activity would cease. And the same thing is true for the maximum. And then you would figure out that window where you get the highest enzymatic activity without having that detriment. also have something called the voluntary thermal maximum. So this is the temperature in which an organism will voluntarily leave the remains. So this particular piece of beef liver has maggots on it. There's a thermal, one of those heat lamps. There's heat lamps from like reptiles on top. And so you'll see these maggots, they're starting to go into proteotaxic stress, and they will end up trying to leave the remains. Otherwise, they end up dying.
Maybe it's gonna take a while.
Some of them end up going underneath too to try and get away from it. And you'll see that too on remains. You'll see maggot masses forming under the body when they usually wouldn't because of proteotaxic stress. Now, at the individual level, there is behaviors that take place to try and combat that proteotaxic stress. So typically it's from that critical thermal maximum, right? That voluntary thermal minimum will be the minimum temperature in which an organism will leave the remains, right? But there's steps before that. So what maggots will end up doing is moving and overturning in the maggot masses or moving to other parts of the body to cool themselves down before reintegrating back into the maggot mass. So the maggot mass will always be hottest at its center. So as the maggots are turning in that mass, they're experiencing different temperatures. So they'll end up having a lot of evaporative cooling. Or they will go into more liquid environments that will allow for some more convection or heat convection to take place. And so you'll have respiration that also happens in the spiracles. So there's posterior spiracles. So it's pointing to here on the end here. There's also spiracles that exist right next to the body. And virtually, like a lot of this is physiology, right? Like the insect is not actually performing any behaviors when they're just doing this evaporative cooling, but they're actively putting themselves in the environment that helps make these aspects a little bit more conducive to reducing proteotaxic stress. One thing I wanted to bring up, because I mentioned in the reproductive strategies lecture about how female flies will always choose a resource to increase their own fitness. Usually when a female is doing this, this is in complete opposition to her offspring's own fitness, because she will lay a higher egg clutch to make sure individuals make it to adulthood. But that also means there's a larger maggot mass, which means that there could be higher metabolic stress or proteotaxic stress that could leave the larvae dead, right? So there is this give and take. And ultimately, when a female does this, they're just giving their offspring more competition. And then ultimately, she's saying, quote unquote, saying, let the best one win type thing. So just food for thought. Now, when you have this overcrowding, right? So again, females are positioned to want to dump as many eggs as they can on a given resource. And because of that, they're not necessarily, there's no thought going into this, right? Like they're not saying like, oh, there's a bunch of eggs here already, I'm not going to lay here. There's not cognition like that. And so because that you have all this oviposition, you're going to have this overcrowding, you're actually going to have a detriment, right? So you'll have increasing maggot mass. It will help with your enzymatic activity and assimilation and all these things until it doesn't. So just like that optimal thermal curve, there is a drop-off point where it no longer is helpful and it's actually a severe detriment. So when we get to that point where it's severely overcrowded, you're actually going to see a reduction in individual growth rates. So where before we would see that acceleration, the body sizes of individuals will be much smaller because there's less food to go around per individual. And so as we talked about in previous videos, when you have a smaller individual, you're going to have less offspring when you're ready to reproduce. So your fitness is lowered. And because of that, you'll end up having that reduced fecundity. So again, smaller body size, less fitness. And then you're going to have increased mortality rates at the individual level. So you're going to have the percentage of your individuals that you start with and end with are going to be much different than those of smaller maggot masses. So a smaller maggot mass might only have 20 individuals, but all 20 of them make it to adulthood, where your largest maggot mass of 100 individuals could only have 20 individuals make it to adulthood. So again, it's not conducive, at least not for the group as a whole. Okay, so that's all I have on the biology of the maggot mass. I will see you on the next video.